Yes. As a bit of context, here I will talk about the global patterns for planktonic marine diatoms using the data generated by Tara Ocean expeditions. And so this expedition was carried out between 2009 and 2013. And you can see from the map that it started in the Mediterranean Sea, then crosses the Red Sea, the Indian Sea, South Atlantic, and then South and North Pacific. The boat passed through the Panama Canal and then the North Atlantic to come back to the Mediterranean Sea. And then after it was carried out a circumnavigation through the Arctic. So it's a nice set covering different type of ocean regions and environments. Different type of devices were on board, but here I will focus in the dataset, the three type of dataset generated. So seawater by a serial filtration system, seawater sample was filtered, and in parallel, what part of the sample was used for the detection by optical microscopy of phytoplankton and different type of phytoplankton, but also other part of in parallel, the same samples were used for molecular analysis. One of them was one of the molecular method implies the isolation of DNA and then using a marker, a taxonomic marker gene, the specific, in particular the 18S ribosomal RNA gene, for analyzing the composition of the community. I mean, this method is called metabarconic and is, let's say, equivalent to optical microscopy because we can obtain the information of who is there, but it has some advantages because it can be high throughput and we can reach deep taxonomic resolution, including species that are not easy to classify by morphology, and also it has high detection power to detect very rare species. And in addition, there was an extra data set that correspond to the analysis of the RNA from those samples. I mean, by metatranscriptomics. And this type of data set can give us information of what are the plankton doing at the moment of the sampling. A bit more detail about this method or the experimental part of this method. So the seawater was filtered, and then using some chemical methods and also physical methods, the planktonic cells are broken, and the DNA and the RNA are extracted, and then they are sequenced, and the sequencing reads are then compared to reference databases for the taxonomic annotations of the reads. And with that, we can continue with the ecological analysis. So a key part of the analysis is that we rely on reference databases. So the quality of the reference databases can limit or not our studies. And this is an important point that I will come back later. So for the metabarcoding of the 18S ribosomal RNA gene, we targeted two type of regions. The genes has different type of variable regions that can be used for these taxonomic studies. One region that was targeted was the B9 region, that is around 130 pair bases of length. And in parallel, it was also targeted the B4 region, that is almost 400 pair bases length. So by PCR, these regions are targeted from the environmental DNA, and we obtain different type of unique sequence that are called amplicon sequence variants, ASVs, that can be used as a proxy of species. Here I am simplifying because it's more complex, but this is a good supposition. And then the fraction of the sequencing reads for a given ASV can be used as a proxy of its relative abundance. So coming back to the data set that is available. So as I mentioned, we have metabarcoding, optical microscopy, and meta-transcriptomics for, in this case, in this later case, for functional studies. And you can see that they cover most of the samples taken by the expedition. Part of the data sets correspond that are highlighted as red cycles correspond to data sets that have been analyzed in a previous study, but for a diatom study, but you can see that many important parts of the oceans were not analyzed that are important for diatoms, for example, the Arctic Ocean. So now I will let's go to the patterns that we obtain analyzing this data set. And for this, I will first focus in the metabarcoding. I mean, the metabarcoding based on the regions before and the region benign of the RNA. So you can see that we can get the relative abundance across the whole ocean and compare with the other eukaryotic phytoplankton to obtain a global picture of the prevalence and diatoms. Before continue here, as I mentioned, the seawater sampling was carried out by a serial filtration system, and in order to enrich different cell size in different size fraction. So you can see that there is a first size fraction that is especially abundant of piconanoplankton and between 0.8 microns and 5 microns or 2,000 microns, depending on the experimental setup that was carried out at that moment. Then another size fraction for larger cells between 3 or 5 microns and NAP and 20 microns, and then between 20 and 180 microns, and finally for very large plankton between 180 and 2,000 microns. So these are the four type of size fraction that was analyzed here. When we compare the relative abundances of diatoms with other phytoplankton at global scale, you can see that diatoms are predominant together with dinoflagellates, but when we integrate the abundances with the abundances, it's clear that diatoms are prevalent, especially in larger size fractions. And in addition, you can see from the box plots that diatoms present the largest abundance variation from very, very low detection to almost the only eukaryotes in those samples. And when comparing in terms of unique sequences, I mean in terms of ESVs, diatoms stand out as the most diverse phytoplankton group after dinoflagellates and aptophytes. There are, of course, certain limitation of these methods for example, there are many ASBs from dinoflagellates that cannot be classified as coming from a phytoplanktonic or a heterotrophic dinoflagellate, but this gives an idea of or confirms diatoms as the most prevalent and diverse phytoplankton group at the global ocean scale. It's also nice to compare diatoms with other traits, such as in terms of against silicifiers. In this case, also diatoms becomes the most prevalent, together with and with an order of resaliers, as the one that is displayed here in the photo. But on its own, this molecular survey is nice to see that diatoms are dominant, not only among phytoplankton, but also among silicifiers. So here I am now showing how is the latitudinal gradient of diatoms. Each road here in the maps, and each road correspond to a size fraction, with a smaller size fraction at the top and the larger at the bottom. And then the first scatter plot correspond to the latitudinal gradient, but in terms of abundance. That is clear that the highest abundance for diatoms occurs in polar biomes. But in addition, for a smaller size fraction, there are some places where they are highly detected, as you can see in the Mediterranean Sea or in the South Pacific. And in terms of diversity, which is the other scatter plot, there is an increase towards the equator. So there is the opposite, as the diversity latitudinal gradient is the opposite of the abundance gradient, probably because well, where diatom communities are very abundant, are hyper dominated by fewer species. So here, this is a PLS analysis regarding the abiotic and biotic variables that can explain the distribution of diatom abundance and diversity. So in the first plot, it's clear that diatom Shannon diversity and diatom relative abundance are correlated with the same environmental variables, but in opposite directions. So diatoms are abundant or very abundant in high chlorophyll A communities, so which is a proxy of high biomass phytoplankton community. The nitrate is a key driver of diatom growth. They are very abundant in where nitrate is high. And also this, when we check regarding the biotic variables, there are some nice patterns. For example, diatoms are abundant, or the abundance of diatoms is correlated with copepods. So grazing seems to be a key factor controlling the diatom population, and they are anti-correlating with the picocyanobacteria and with some silicifiers, or at least with the most abundant silicifiers. So it seems to show some competitive interaction over silica, and the anti-correlation with picocyanobacteria maybe can be related to what it is known regarding the out-competition of diatoms in nitrogen limit environments by cyanobacteria, and in these environments, in general, they are low chlorophyll levels. So this all these patterns are related. And regarding the patterns across size fractions, the highest relative abundance of diatoms was in the nano was among nanoplankton. I mean, in the size fraction between 3 or 5 microns and 20 microns in that size range, in which we detected a mean of 90% of eukaryotic roots correspond to diatoms. And when we check the environmental conditions associated to the different diatom size, we see that larger diatoms are associated with high latitudinal regions rich in nitrate, and smaller, smaller diatoms here in red or also in blue are prevalent in more oligotrophic regions and temperate waters. Well, the diatoms, as you know, can be classified in 4 main classes. Um, um, we see the prevalent in the at global ocean level, um, we and in planktonic lifestyle were Mediophyceae by far, and then following by Raphid penates and Coscinodiscophyceae. Araphid penates were very low abundant and only prevalent in an Arctic ice age station. This is expected given that they are more prevalent in benthic environments. And in terms of environmental drivers, rapid penates here in dark green are predominant in temperate environments, although also are significant at some high latitude regions, as can see from the maps, whereas Mediophyceae and to a lesser extent Coscinodiscophyceae were highly abundant in high chlorophyll and high latitudes. And then there was also a clear size partition between the different classes, with Mediophyceae very abundant in intermediate size fractions. At some extent also Coscinodiscophyceae, but Raphid penates shows a clear decrease in abundance towards larger size fractions. And then we focus in the top most abundant genera. They account more for this for most of the signal, the molecular signal. In particular, they account for 92% of the assigned reads for V9 beta barcoding and 90% for V4 metabarcoding. And you can see, well, Ketocero, Thalassiosira, Actinocyclus, and Pseudonistia were the most prevalent by far. And here in the graph, you can see the whole list of the top 20 most abundant genera. In the first column is the rank according to the V4 dataset and according to the V9 dataset. Even though there is some debate has been some debate in the community regarding which regions are better to target diatoms. Our results are nice in terms that they seem to be they seem to be very similar, and the methods are robust enough so that there are 15 common genera in both datasets, And in addition, there are many other six genera that are in one dataset as top 20, but still they rank high in the other dataset between, let's say, positions 21st and 28th. The few cases in which there were strong inconsistencies between or discrepancies between datasets, it corresponds to Fragillaria, Asteromphalus, and Progmaniella that were only detected in the B4 dataset, and Thieres that was mainly detected in the B9 dataset. This can be related to the reference databases, the lack of enough sequences for these genera in the reference databases, as well as to the difficulties for the amplification by PCR of these regions in these specific cases. For one of the size fraction, we have curated microscopy datasets. In particular, this size fraction correspond to the one between 20 and 180 microns. So we also compare the top 20 most abundant genera between microscopy and the two types of metabar coding. And here also we obtain high similarities between the different methods. In particular, 13 of the top 20 genera identified by microscopy were also present among the top 20 in both molecular datasets, and 3 additional genera from the microscopy top 10 are found among the top 20 in only one of the other molecular datasets. And again, here for the explanation, we have some limitation in the molecular techniques in terms of lack of molecular references in reference databases. For example, in the case of Cylindrotheca and Thalassiocinema. And on the other hand, we have also the cases of the difficulties in identifying by microscopy some other genera, such as Minidiscus, Arcocellus, or Aslea, genera that are small or not with not distinctive morphology. And well, so after that, we also analyzed the biogeography of the top 10 most abundant genera for each molecular dataset. Here the maps are showing the tendencies merging both B4 and B9 metabarcoding datasets. And well, these are the patterns in which we find some genera like Etocero, Thalassiosira, Actinocyclus, and Fragillariopsis that, or to some extent Fragillariopsis, that are cosmopolitan, but much more abundant in Arctic and Southern oceans. Then other genera that is, or other genus that is cosmopolitan is Pseudonychia, but in this case is more homogeneously distributed. In the cases, and so there is another big group of genera that are mainly detected or almost exclusively detected at high latitudes. In the case of Forocera and Proboscia in both Arctic and Southern oceans. In the case of Fragillaria in the Arctic Ocean, and for Corythron, Trieres, and Hemiaulus in the Southern Ocean. And finally, we have the cases of Guinardia and Leptocylindrus that are detected in tropical and subtropical regions. So in addition to this analysis by separating diatoms according to classes or according to genera, we can also analyze all the ASB together, or all the lineages together, and do some type of clustering analysis in order to detect communities of co-occurring diatom lineages and analyze how these communities varies in the different under different environmental conditions and different ocean regions. So for both before and benign dataset, we were able to define 25 different diatom communities. And you can see here in the lower panel with different colors that they vary in terms of biogeography and also in terms of environmental preferences. But something that is very clear is that here in red, in the lower, the last row in red, the red corresponds to the Arctic Ocean. So you can see that there are some communities that are very prevalent in the Arctic Ocean. And so the Arctic in itself stands up as a hotspot of diversity and abundance of diatoms. And one important part of the dataset, or something that to have in mind, and that is very interesting to go more deeply, is those sequences, those ASBs that we are not able to assign at the level of genus or species. The unassigned ESBs correspond to more than 41.5% of the total V4 ASBs and 57.9% of the total V9 ASB. In terms of abundance, I mean in terms of reads, the proportion is lower, 25.7 and 19.2 for V4 and V9 respectively. So this means that we are detecting many ASBs, many diatom lineages that we cannot assign at the level of genus or species, and they are rare diatoms. Well, and then when we check in terms of let's say the distribution of unassigned reads in the different size fractions, there is the higher levels are in the among the nanoplanktonic diatoms. I mean between 3 or 5 microns and 20 microns, which makes sense because more traditional studies are or the detection based on detection of microscopy are more focused on larger size classes. When analyzing the regions in which unassigned reads are more prevalent, we have a higher relative abundance in the southern hemisphere, including the South Pacific Ocean, the South Atlantic, the Arctic Ocean, but in particular the Southern Ocean. A great proportion of the reads from the Southern Ocean are unassigned. That means a mean of 10% of the eukaryotic reads in the before database. The data set from the Southern Ocean are unassigned, for example. So there is still a high amount of biodiversity that we need to study more to obtain more clues about them. So far, I have been talking about the analysis of the diatom communities in terms of taxonomy and taxonomic composition, using metabarcoding and also in comparison with microscopy. But now I will show some results regarding the analysis of the metatranscriptomics datasets that, as I mentioned, we are capturing here the levels of mRNAs, and thus this is a proxy of the functional activities that the diatom communities are doing at the moment of the sampling. And for this, here I am displaying the list of top 100 most abundant transcript families at global ocean scale. How are they distributed according to the different ocean regions, and the distribution according to their size fractions. So I want to, for the sake of time, to highlight a couple of cases. For example, light harvesting complex, which are the chlorophyll A-B binding proteins, are the most abundant ones of these families, with 3% of the total metatranscriptomic signal or abundance corresponding to this family. And then among these all these top 100 list, there is only one family in which we don't have a clue about their function. The family is named DAF 285, DAF for a domain of unknown function. So these are two interesting cases to go more deeply in the analysis. So in the case of light harvesting proteins, this is nice because a great part of the transcriptional effort is focused of the diatom community is focused to express these families, and these families cover many subfamilies that are involved in completely opposite activities. Some of the subfamilies that are called the subfamily F, Q, and R are involved in light harvesting, whereas the subfamily X and Z are involved in photoprotection. I mean, in heat dissipation of the excess of light in order to protect the cells from and fine-tune photosynthesis. So these subfamilies show different type of patterns according to different environmental partitions. For example, photoprotection families are more expressed in the surface of the ocean, whereas light harvesting is more expressed at a deeper depth. I mean, at the level of the deep chlorophyll maximum. There are also some partitions in terms of size fractions with light harvesting families, like the families F, expressing with higher expression in towards larger size fraction, whereas photoprotection families decrease their expression or among the pool of transcript towards larger size fraction. And this is related with a process we can, let's say, we can think that this is probably related with a process that is called intracellular light attenuation, in which as the cell volume of the diatom increases, the path length of light within the cell also increases, so the light intensity per unit volume is reduced. So this is also nice to see some patterns based on taxonomy across size fraction that can be also at some moment associated with functional patterns, as in this case for light harvesting complex proteins. And finally, in the case of the enigmatic DAF 285, it's interesting to see that the expression of this family is very abundant at high latitude. When checking the 3D structure of the protein encoded by these genes and transcripts, they showed what is called a solenoid conformation. And by this, I mean that these correspond to proteins that are made by small repetitions. There are many of these type of proteins that have been associated with antifreeze activity. So this is a hypothesis that we would like to explore more. But well, so in conclusion, here I want to highlight that we can obtain different conclusions from the point of view of methodological information, but also the point of view of diatom community and diatom functional diversity and functional acclimation. So we obtain a high methodological agreement between the two different molecular approaches that we use for metabol coding, the targeting the B4 and B9 regions of the 18S ribosomal RNA gene. So there were 15 genera that were common among the top 20 in both B4 and B9 datasets. And this was also corroborated by optical microscopy. We, let's say, confirm the importance of diatoms at global ocean scale by as a key or very abundant and diverse photosynthetic group, and also very abundant silicifier group, and with prevalent genera such as Ketocero, Thalassiosira, Actinocyclus, and Pseudonychia. We also were able to define 25 different distinct diatom communities that show different marine environmental preferences. And in this analysis, we obtained 6 communities that were unique to the Arctic Ocean, which illustrate this region as a hotspot of diatom abundance and diversity. And also the fact that we find many of the most abundant diatom genera to be prevalent in the Arctic. Finally, we obtain some functional insights. For example, the increased expression of certain proteins in high latitudes, as it's the case of DAF 285, which is an unknown protein, and this give us ideas and hypotheses to determine its function in the future. And also we found some expression patterns related to death and cell size in the family of the light harvesting complex proteins, which are the family of genes in which diatoms invest most of their transcriptional efforts for their expression. So I want to thank all the people that were involved in this work, as well as the funding. And of course, I am more than welcome and open for questions.
Hi, Juan. Great talk. Thank you very much. It's very interesting. If anybody has any questions, feel free to drop them in the chat or raise your hand, and you can ask Juan yourself, which we prefer, that sort of interaction rather than me reading your question. But I'll maybe jump right in with a question while others formulate theirs. When you spoke about diversity in the diatoms and hotspots of diversities, you mentioned that there were some areas that would be dominated maybe just by a couple of genera where the diatoms were really prevalent. Did you see a lot of high ASV diversity within those areas? So like maybe is there this intergenera, interspecific diversity where the diatoms were really dominant?
Yeah, that's a good point. No, I haven't checked intra-species diversity. And another point also that at some moment we were willing to do in the future is also intragenomic diversity, because that is a source of we can be overestimating diversity due to that effect that also affect other phytoplankton in addition to diatom. But this is a good point to analyze in the future. Yeah.
Great, thank you. Hey, Matt, jump in there.
Hey, Juan. This was a really interesting talk, particularly the molecular ecology stuff. I thought was really intriguing, and there's a lot of room for growth there. Without without getting too far into the weeds here, I am curious about what you've just been mentioning about particularly genetic diversity within and between genera. You mentioned that there are some obvious blind spots in the reference database. And so I'm kind of wondering if sort of what the general practice is that you guys are using to define your genera within the species data and the ASVs.
Yes, we are using we can define, let's say, use one possibility to use a sequence identity cutoff and consider to above the cutoff, we consider that species or genera. In our case, we are using another way of assigning based on the program Data2 that gives some probability based on some training of reference sequences. It can try to learn the different cutoff to different genera. Of course, always taking the reference database as the truth. And using some method, they can assign different type of genera, but using a mobile cutoff that seems to be seems to work better than just using an arbitrary cutoff, at least when comparing with microscopy data.
So those thresholds are evolving and they are being updated.
They are not fixed. It will depend of the species and the genera. The main issue is that we are of course biased by the reference database in terms of the sampling that is representing or the representation in the reference database, and sometimes also by contaminations and errors in the reference database. Yeah.
Right. Yeah, I mean, just stuff like I noticed you mentioned Guinardia particularly in some of your distribution studies, and I mean, there are Guinardia taxa in the Cosenodisci, there are Guinardia taxa in the Mediophyceae. So, obviously, keeping those reference databases updated is really important.
That is super important, and fortunately, the PR2 database is been updating very frequently, based on the one that is mainly used for marine studies, and also curated. And so in that case, this is super important. But yes, this is something that is evolving and improving as much as we can. But there is, in the case of then moving to the other issue of metatranscriptomics and functional annotation, that the limitation of the database is even more problematic, because it's more difficult to and costly to sequence a whole transcriptome or a genome. So that's a bigger problem even. Yeah.
Okay. Cool. Thanks.
Looks like Anna has a question. It's interesting that the Fragillaria is so abundant in the Arctic. She's never seen lots of it there. Could it be Fossila now, or Fossila ficus? Perhaps it's just a taxonomic discrepancy in the database.
So let me check. So she mentioned Fragillaria, no?
Yes.
Yes. Yeah, that was a surprise for us. As we consider it to be mainly benthic genus, but this is a clear. This is a nice an station that is very close to the ice. And also when we check with the microscopy data of that sample, the morphology seems to be Fragillaria. So maybe it's something specific of that sample. But of course, yes, it will be. It's something that we have been. It has been in our minds, and we would like to go more deeply with the analysis. But yes, when the microscopy data was what give us some support to believe that is Fragillaria. Yeah.
Sarah, you have a question?
I do. Yeah, Juan, thank you for a terrific talk. This is just really fascinating. And at the beginning, you talked about that these samples are also worked on in light microscopy. Are those materials archived for future work?
I think so. I mean, to give you more context, this is was carried out by a group in Stazione Zoologica di Napoli, in Italy, by Adriana Zingoli, mainly carried out by Adriana Zingoli there, and I'm not sure if regarding the question, but in addition to this optical microscopy, there has been also taking, let's say, on both, they have been taking some other samples for high throughput confocal microscopy, and the images are publicly available, and we have done some stuff also with those data sets, and that can be also a complementary data set to analyze. And that data set is still under use, so it's nice for the community to know that it
Excellent.
exists.
Yeah, that's great. Maybe I'll follow up with you about how to find that publicly available.
Yes, I can, let's say, share very quickly let's see.
Yeah, and just in terms of the archiving too, or just even between the light microscopy, resolving the reference libraries, is that an effort at all to refine the reference libraries based on microscopy, as well as learning more about the ASVs?
Yeah, that's a good point. We didn't. I mean, we went more. In addition to this comparison, as the one that I'm showing, then we check some specific cases, like in the case of Fragillaria that stood out. But then we didn't go more deeply to link, let's say, morphologies and molecular data that can be used to improve, for example, the reference databases and so on. And that is a nice analysis to do in the future. I know that other people in Tara what are they doing is doing some single cell isolation and targeting sequencing the ribosomal RNA and also taking picture of the diatoms in order to make in the future a link reference database of both morphology and sequence, and that's also a nice way to analyze. But already with this data that is available, as far as I am aware, still nobody's doing a deep analysis to try to link, yes, molecular and morphology data. Yeah. Very deeply.
And if no one has, oh, Leila has a question.
Oh, thank you. I wanted to just to ask you, when you compared surface sediment diatom occurrence and the maximum chlorophyll depth occurrence, sorry, you said that it's only cell size. But what about the ratio between diatom and dinoflagellate, for example? And in the high latitude where you have your unknown DUF 285, do you have some other dinoflagellate or other things with it? Or, I mean, the competition in between these two phytoplankton. Thank you.
Yeah, thank you, Leila. So let me know if I put it. So you are mentioning the interaction between diatom abundance and other the abundance of other groups.
Dinoflagellate, and as well the comparison between surface sediment sample and the maximum chlorophyll depth.
Okay. Yeah.
Not only with the cell size, but in between competition in between.
Yes. When I did a comparison between surface and deep chlorophyll maximum, and I didn't find, in general, in terms of at least from relative abundances, for diatoms were more or less the same in the data. There are some concerns because the the DCM in some cases maybe they were not exactly the DCM, so there were some discussion about that. But yes, I didn't find at global level changes in the relative abundance of diatoms as a whole. And then when checking specific genera, some of them were more prevalent in the surface, but most of them were equally distributed in DCM and surface. And then regarding the, let's say, interaction with dinoflagellates, I didn't analyze them. Maybe it could be a nice, maybe I can include this in this PLS analysis of biotic libraries variables, also dinoflagellates, to see if I notice a pattern. But what I can say from what I remember is that yes, there were a clear, let's say, in regions that in which diatoms were decreased, the abundance of diatoms in many regions, the dominant become by far dinoflagellates. But yes, I don't have more analysis in mind or patterns in mind to mention something interesting.
Okay, and for your unknown species, does it occur alone or with some other? the unknown DUF 285, does it occur?
Ah, okay. Sorry. So yes. Yeah. So this I check the distribution of this, the taxonomic distribution also this protein, and it's found mainly in marine phytoplankton and in a few viruses for marine phytoplankton. And the only exception beyond those taxa is Mycobacterium. Is the only exception. So, and then when I check the distribution in the I think here.
There
So
was
I
no
want to check
any sorry,
yeah.
no Rhizosolenia with it?
You mean if I check at the level of the different genera,
Yeah.
how
The
is
association.
it distributed? No, I haven't checked that. Or I think I checked, but a long time ago, I don't remember. What I did is checking not only in diatom, but also in another phytoplankton groups.
Okay.
And what I saw is that this latitudinal gradient of higher expression, but not only gene expression, but also gene copies towards the high latitude is happening also in other phytoplankton groups. So it seems to be a conserved strategy. But yes, I think that it would be good to see how they are distributing among diet. What I did, now I am remember that I also check the copy number variation in different genomes that were reconstructing from the DNA from the same samples as this analysis. It was particularly more abundant in certain species, isolated, more abundant in the high latitudes, and they correspond, there were a few genomes from Fragilariopsis. There were not Rhizosolenia. And yes, I don't remember the other species from the list. Yeah.
Okay, thank you very much.
All right, I'm going to give a warning if anybody has any last questions. I'm going to ask my last question to Juan, and if you have another question, please throw it in there or let us know. Juan, thank you so much for fielding these questions. Really love your explanations and your talk overall. If you could, you've been going back and forth in your slides. Could you go back to the biplot of the abiotic and the biotic one more time? And I was just curious if you had ever analyzed the biotic variables with the kind of the coarse diatom morphological groups broken out, looking at the raphids and the pinnates, because I work in the Great Lakes, and while we're not in the ocean, sometimes I will see these raphid diatoms out in the middle of the water, and I say, what the heck are you doing out there? Why are you spending all this effort making a raph and living out here? And I was just curious if the raphid diatoms maybe were, living with maybe some other filamentous algae or colonal algae.
Yeah, I see. Well, that's a good idea to do with the data. Yeah. Frankly, I haven't done it. But yes, I can try this day and let you know if there is something interesting. Yeah.
Cool.
So far, the only analysis somehow related with that was the analysis of the classes, let's say, but only using contracting them with abiotic factors. So yeah, that's a good point to analyze. Thank you, David.
Oh yeah, sure. I'll stay tuned. I look forward to hearing the results. I have one final wrap-up question. It's actually for Sarah. You mentioned you will be potentially streaming some of NADS. Will we be able to find that on diatoms.org?
Yes, stay tuned to diatoms.org. I hope to have something later today to list their 4 plenary talks, and they'll be at unusual times. And we're going to do our best to stream those plenary talks and also record them. You don't have to do it, David.
Okay, great. No, I wasn't worried about that, but just making sure folks could get the information if they couldn't attend.
Yeah.
But Juan,
And also
yeah,
people,
go ahead, sir.
I'll just say people were asking about your publications, Juan, and I will be sure to put links in the page where we have this talk, and then David will also load the recording.
Perfect. Thank
Thank
you.
you so much, Juan. This was really
Thank
great. Really appreciate you.
Thank
Thank you, David. Thank you, Sarah.
Have a good day, everyone.
Thanks everyone for joining. See you next time.